Any questions? There's one.
I have two questions. So my first question is about the ASO modification, and both Jeff and Nadina, sorry, mentioned in your presentations. So there are various types of modification, including like LNA and MOA, or MOE, or GAPMER. So I want to hear your perspective or rationales how to choose the modification, pros and cons for each different type. So that's my first question, ASO modification. The second question is more for Nadina. So would you mind to elaborate a little bit more regarding how Lauren's team access the off-target effect for your ASO hits?
The regulatory experience, at least the feedback that we're getting from some of the ex-US regulators, is the commonality of backbone is something that they would actually really like to see if they're going to approach this more like a platform approval. It doesn't mean every drug just gets automatically approved. It just means they can truncate the amount of data that's needed for the review. And they've at least signaled early on that things like LNAs, which does create better stability, they actually mentioned that in their feedback to us that they would rather not have different backbones, that would require a different level. So we're somewhat erring on the side of let's just get through what we can, even though I think we would really enjoy the opportunity to put an LNA in there. But that's just kind of one example where their feedback is that the commonality of backbone gives them more of a predilection to say, yeah, this is a truncated review based on the amount of repeatability you can show.
Yeah, I'm just going to add to that, that essentially there are a lot of years of experience now where we kind of have nailed, well, I'm going to use the word nailed, but I'm not sure if that's the correct word, the chemistry for GAPMERs and also for splice switching ASO. So for example, essentially for that, we know that for research purposes, so if you're in a lab and you want to use LNAs, that's completely fine. For clinic, we do know that these modalities are a little bit more toxic. So in the terms of the backbone, phosphorothioate is something that we routinely use, and especially in a GAPMER, I'm going to go technical here, in a GAPMER for CNS, and make sure our phosphorothioate with a phosphodiester is the one that we have the most knowledge on and the most results on. 2'-methoxyethyl modified on the RNA. This is also something that we have a lot on. And, also the splice switching, and Jeff can elaborate more on that, that is fully modifying an 18-mer, and essentially the backbone is mainly phosphorothioate. This is also something that has been now well established. So I think that they are, and the reason, as I was saying, that we use the chemistries is you want a drug, an oligo or whatever, to last long, to have less immunogenicity, right? To have high affinity for your RNA, which is essentially your target. And lastly, you want the best pharmacodynamic properties. So all of this together, this is basically what is on your tick list in order to define the best chemistry. And these chemistries are pretty much now defined at least for the ASOs.
Yeah, I mean, 250,000 people dosed with that chemistry. So that's a platform that has been tried and true. It's tested in many ways in the clinic. So that's basically it. That's where we focus. And at Ionis, we did a lot of work around, understanding the biology first and then teasing out the chemistry if we have to. So the idea was to find the target space, and then if you have to do a chemical SAR around a particular spot to tease out toxicity or improve potency, then that's kind of the strategy we took. I mean, for rare disease, it's just the clinical proof of concept. What's the fastest or the cheapest way to kind of get through the path? And if you change that chemistry, you throw a big wrench into the equation.
Okay. More questions here?
The question for Adina or Jeff. My understanding is that this, for ASOs, the delayed tox is something that's, it adds a lot of time to to the development, but is also a little bit hard to predict from the sequence. Do you have any idea, what may be leading to that, and are we getting towards, some predictiveness for that?
It's a great question. It is a really good question. So I think that, and I'm going to share my perspective, there are twofold to this question. There are aspects of it that we can predict, and there are aspects of it of like some motifs in sequence that, you can build into your algorithms and predict that some of the things could potentially cause toxicity. And especially you would see that in the tolerability, which is, a smaller scale tox study before going to GLP tox. So that is something that, the more learnings we have, yes, we can fish some out and just, either cytotoxicity or immunogenicity. These are the two main things. But there are also some other aspects that at the moment we cannot predict them. So at the moment there is a molecule that can go to tolerability. And that's why I think from my perspective, these toxicology studies are very, very important. Yes, they're costly and they're lengthy, but this is why they're important, because at this moment in time, there are some that we can predict, but there are some that we think that is not immunogenic, is super safe, not cytotoxic. I can't see anything wrong with it. Cells tolerate it just fine. And then we go to rats, and then they are either, acute FOB or delayed FOB. So there are things that we cannot predict. There are things that, and at least at Lorem, we really try to look at the data and see for any commonalities, because obviously having less of these leads into tolerability, we don't want to lose any good drug. But at the moment, this is a work in progress. So some we can predict, some not.
Yeah, and I'll just add to that it comes down to the data. I mean, there's some really good models that Ionis has developed, and but having delayed tox data is harder and longer and more expensive, and that's really what it comes down to. Once that data is there, then yeah.
Okay, we have two questions from the outside. The one is a more general one. Is there a centralized database that collects all the genetic data from rare disease patients around the world?
Unfortunately, no. We don't even have a centralized database in this country, but there are a number of countries across the world that are putting in, I would say, yeoman's work to try to create databases that are largely newborn screening. The Genomics England database is, I think, well over 100,000 at this point. Emirati and the Qatar genome projects, actually, I think Emirati alone is almost 1 million full. In Qatar, I think, is somewhere around 250,000. There's about 4 or 5 states, I know California is one of the states, where there's upwards of tens of thousands of newborn screening with whole genome sequencing, but there's not even a database in our country to do that. And I think, as I maybe alluded in my talk, wow, that is a fantastic effort for us to do for so many different reasons. And I think it opens up an entirely new ability for us to target disease. There's going to be issues with that too. I mean, one of the things that I noticed at Arab Health just a few weeks ago in Dubai is that they are incorporating newborn screening with genetic counseling and premarital counseling as well. And that's a concept that is going to be something to tackle as we get closer and closer towards having this evidence of gene and the number of mutations people may carry.
So if I can comment, there are, is that okay if I comment? I don't know if it's.
Yeah, absolutely. Go ahead.
There are, the issue is with data privacy, obviously, and different countries handle data privacy very differently. The Scandinavian countries have really good genetic databases, as the countries that Kent mentioned. But for example, if a company wants to use them, they have a very hard time accessing this information. If only mutation information is needed, then ClinVar is probably the best source for genetic information that's totally de-identified and just has information about mutations in genes.
Okay, thank you. I have one more question online, and then maybe one more in the room, and then we have to move on. Or two maybe. So one that came in online is: are there any challenges for ASO administration that goes intrathecal?
Yeah, I mean, there's always going to be challenges anytime you're in that space. I'll just say from a research perspective, it's challenging from the perspective that on the one hand, it's hard to find people that can actually do it well and do it well and consistently. So if you think about just actually doing that in a research setting and the consistency is a challenge, think about it in an actual treatment setting as well. And that's always going to be the concern with any kind of intrathecal administration. So the idea of accredited treatment centers, places that actually are defined in being able to do this, there's multiple disciplinary aspects of those treatment centers. What we see in research is what we're going to see in the real world settings as well. But it's not an ideal space. That's where, for me, I mean, we're intrathecal administration, of course, but I'm really hoping that there are companies that are going to come along with different modalities of delivery. I think it's going to take a while, though, unfortunately.
Yeah, and just to add to that, that's why I was saying that having really, really good physicians and really good treatment centers, as you were saying, that they're really equipped. I mean, of course, you require anesthesia a lot of times because of the specific challenges of these patients and dosing them intrathecally. However, having said that, it is a route of administration that is well established, and unfortunately at the moment, until we have another, exactly, and a safe one, and until we have another delivery, this is.
We have one more question.
Yes. Thank you. Well, first of all, thank you all for the session. It was fantastic. So my question is on behalf of my husband, Bernie Ziprich. So Kent, you talked about the need for a better regulatory pathway, but Enlorem seems to be finding success with the current FDA framework. Can you help us understand the nuances better and some of the changes you're hoping to achieve?
Yeah, it's a great question, because I think in some respects, folks may see as Enlorem and Everyone Medicines and others as sort of in competition. We don't see it as competition. I mean, I think the one stark difference between what Enlorem's doing versus Everyone Medicines is that we're actually just going down the route within Europe to create a brand new pathway. And I think Enlorem is actually going with the existing pathway, but using that totality of data to also negotiate a new pathway while going down the existing one. Our attempt is to really try to create as much data as we can in this sort of compendium approach. And the thinking behind it is that the timing right now, at least in Europe, seems to be very favorable. They clearly understand you can't do a clinical study. The question of GLP tox, quite honestly, even if they require that, if it's a GLP tox that's reasonable and that we can figure out a way to make it even somewhat affordable, that's okay. But right now, there's not a pathway that says it's not only approved for marketing authorization, but then it'll get reimbursed as a result. I think we're all kind of trying to bring as much as we can to bear to give them the experience to say, yeah, this is how to do it. And this is the way to go about it from a research perspective. And it's not just for research, it's for treatment and approval. Reimbursement's the key here.